It's my pleasure to introduce Dr. Susan Weiss today to our seminar series, and she's really quite gracious to join us again in Boston and Cambridge. She was here right before we shut down. I just have this memory of it being our last Harvard virology seminar before the shutdown. And so thanks again for joining us. So Dr. Weiss obtained her PhD in microbiology from Harvard University, working on the RNA paramyxovirus Newcastle disease virus with Michael Bratt. She then headed out west to UCSF to do postdoctoral training with Michael Bishop and Harold Varmus at UCSF, studying the avian sarcoma virus RNAs. And she then moved to UPenn, where she had the foresight to focus her own research program on coronaviruses, opening her lab in the very year of the first International Coronavirus Symposium, which she mentioned in a nice piece recently in Journal of Experimental Medicine, had about 65 people in total present. I'm sure that'd be quite different at the next one. She's now the professor and vice chair of the Department of Microbiology and the co-director at the Perelman School of Medicine, where they have a center for research on coronaviruses and other emerging pathogens. She's worked on many aspects of coronavirus replication and pathogenesis over the last 40 years, making contributions to the understanding of the basic biology, organ tropism, and virulence, working on the mouse coronavirus MHV68, the MERS coronavirus, and more recently the Sars coronavirus 2. And so I think we'll hear about her work that over the last 10 years has focused on coronavirus interaction with the host's innate immune system and viral antagonism of double-stranded RNA-induced antiviral pathways. So thank you again for joining us this morning.
Okay, so my talk's going to be divided into two parts. The first part's going to be the history and biology of coronaviruses. And I think it's important to understand the biology of coronaviruses because it allows us to identify steps in the virus life cycle that may serve as targets for antiviral therapies for most, if not all, coronaviruses. The second part of the talk is going to be on innate immune antagonism of coronaviruses. And I think this is important because it's pretty clear that during early infection of both animals and humans, both MERS and SARS really failed to induce very much type 1 interferon, type 3 interferon as well. Whereas during the later infections, the production of cytokines like interferon as well as other ones can be pathogenic. Okay, so coronavirus, for those who've heard my talk before, there's going to be some redundancy, but there's going to be some new stuff as well. So coronaviruses are members of the nidovirus family that's shown here. Their nidoviruses are named for their nested subgenomic mRNAs, nido meaning nested, generated during infection. They're all enveloped, kind of medium-sized viruses, and they all have single-stranded positive sense RNAs. And here's a blow-up of a coronavirus, and these are the now familiar, I think, spikes that form the corona or the crown-like morphology that's exhibited by coronaviruses. This is the virus particle. It's really pretty simple. There's this very long RNA. It's about 31 kilobases, the longest RNA viral genome that we know. It's non-segmented. It's capped and polyadenylated, so it looks like a host mRNA, and it actually does serve as an mRNA, as we'll see later. It's complexed with a basic protein, nucleocapsid protein, and forms this helical conformation. Around this nucleocapsid is the membrane, which is derived from the host cell. It has 3 viral proteins in it, the spike protein, the now famous spike protein, that's really important for entry as it binds to the receptor and induces viral to cell fusion. It's important determinant of tropism, immune response, virulence, and now we see also for vaccine development. There are 2 other proteins in the membrane protein M and the small membrane protein E. These are both important for viral assembly and also do have functions during the virus life cycle. Some coronaviruses also encode another protein called hemagglutinin esterase, which is kind of analogous to influenza HA. This is not expressed by SARS-like viruses. The coronavirus timeline. As Ben alluded to, coronaviruses that started really, there's research back into the 60s and 70s, but really started around the late 70s and early 80s. So I call this period the period of studying mouse hepatitis virus, the murine coronavirus as a model. Other animal viruses, there was a lot of research done on like IBV chicken virus, bovine coronavirus, porcine coronaviruses, often from a vaccine perspective. And there was also some work done on the human cold viruses, OC43 and 229E, which are now much more well known than they had been. They both cause the common cold. OC43 can also infect the lower respiratory tract. And I found papers on these viruses way back into the 60s and 70s. And then things were pretty quiet in coronavirus research until 2002. When SARS emerged. But during this period, there was a lot of work going on these viruses, the MHV model and the animal viruses, that a lot of the molecular biology that I'm going to talk about, how the virus enters the cell, how it replicates, its receptor, just really a lot of work done on the very interesting transcription and translation mechanisms that these viruses use. As we all know now, I think SARS originated in bats, transmitted to civets and then to humans. It caused an epidemic, mostly in Asia. 8,000 people were infected. It had a 10% mortality rate, but the epidemic was pretty much gone after 8 months or so. But two things that happened after that I think are important to note. People looked for other coronaviruses and other human viruses and identified HKU1 and NL63, causing pneumonia and bronchiolitis and croup. And these are really the only human coronaviruses we know of other than the really lethal ones. The other thing that happened is it became apparent that there were a lot of coronaviruses in bats, some related to SARS, some not. And then things were kind of quiet until 2012 when MERS evolved. MERS emerged in the Middle East, causing another epidemic, which is actually still going on. There's still people being infected by MERS, not that, maybe like 2 a week from what I understand. And MERS emerged from bats into camels, where it actually has a reservoir. So it's very common to find MERS positive camels, both in the Middle East and in Africa. So this, and MERS was not quite so contagious from person to person as SARS. So MERS still kind of percolates along. And then things were kind of quiet until late 2019 when SARS-CoV-2 emerged again in China, this time in Wuhan. This was in South China. And we all know what happened. That's why we're here today. And I call this sort of the pathogenic human coronavirus era. We have SARS-CoV, MERS-CoV, and SARS-CoV-2. They all cause severe respiratory disease. And just to distinguish, SARS-CoV-2 is a virus that causes coronavirus disease 2019 or COVID-19. This is kind of analogous to HIV and AIDS. Okay, so just as Ben mentioned, the first meeting that we had was in 1980 in Würzburg, Germany. About 60 people were there. That I think was most of the field. In 2003, after the first SARS emerged, there was the 10th international meeting in the Netherlands, and there were hundreds and hundreds of people there. And there was supposed to be the 15th meeting, they're now called Nitovirus meeting, in the same place in the Netherlands, which is a little bit eerie, in 2020, and it was postponed until next year. I hope that it can meet. Okay, so let's go through the coronavirus genome structure. Human coronaviruses are found in all these different lineages. The alpha coronaviruses include 229E and NL63, and the beta coronaviruses. There are 4 lineages, but 3 of them contain human viruses, lineage A, B, and C. These other names are the new names, actually the correct names now for coronaviruses, although they're kind of cumbersome, so I'm going to stick with A, B, C. Among the lineage A viruses, there's OC43, HKU1, and murine coronavirus that I'll talk about a little bit because that's what I've worked on a lot. Lineage B, SARS coronavirus, and lineage C, MERS coronavirus. So all coronaviruses have this very long replicase gene that encodes 16 non-structural proteins that I'll talk about in more detail. It's about 20 kilobases. The rest of the genome contains spike E, M, and N genes in the same order for all the different lineages. But what makes these viruses, these lineages different from each other is these 3' ends. These numbered open reading frames encode small proteins, usually oftentimes interferon antagonists. And that's what, to me, gives each of these lineages its own character. So soon after SARS-CoV-2 emerged, we learned that it was very close in sequence to SARS and had this similar genome organization, and that's why it's called SARS-CoV-2. Okay, I want to go very quickly through the very simple model of coronavirus replication, which we drew in 2005 after the first SARS emergence. There's things missing that we know about now, but very simply, the virus attaches to its receptor via the spike protein. The viral membrane merges with the cell membrane and the nucleocapsid is released into the cell. Nucleocapsid, the RNA and the nucleocapsid is transcribed into a polymerase, which then transcribes these subgenomic mRNAs and replicates genome RNA, shown here. The viral membrane proteins inserted to the cytoplasm, into the intracytoplasmic membranes here, M and S, ME and S. And the nucleocapsid protein assembles with new genomes and buds into these membranes in the ERGIC, the endoplasmic reticulum Golgi intermediate compartment. And then we see virus particles and vesicles being transported to the cell surface, where they're exported into the extracellular space. And meanwhile, the spike proteins are dotting the membrane along here, and they cause cell-to-cell fusion, which you'll see later. So I want to go through several steps in the life cycle that I think are really conserved among all coronaviruses: entry, genome RNA synthesis, and translation, and all the conserved replicase proteins. So viral entry is pretty interesting, I think. There are two routes of entry into the cell. The first route is by the direct plasma membrane route. By this route, the viral spike protein attaches to its receptor, which in the case of SARS and SARS-2 is angiotensin converting enzyme, or ACE2. It's then, the spike protein is then cleaved by a cellular membrane-associated protease. In the case of the airway alveolar 2 cells, it's TMPRSS2, and that allows the spike to change conformation and trigger a fusion between the viral membrane and cell membrane, releasing the nucleocapsid into the cell. The other route is an endosomal route, whereby again the spike protein attaches to its receptor. This time the virus is internalized into endosomes, and in the endosome, the spike protein is cleaved by cathepsin, which is a low pH dependent protease. And then again, there's a fusion between the viral and cell membrane, and the virus is, the nucleocapsid is released into the cell. So the big question is, what determines which pathway is used? And that's a combination of the proteases available. We have the membrane-bound protease TMPRSS2, the cathepsin, the endosomal protease cathepsin. We also have there could be other membrane-bound proteases. There's also furin, which everybody's heard of, I think. Furin is an intracellular protease that cleaves the spike protein during viral assembly, so that when it comes out of the cell, is already cleaved by fusion. So let me give an example of how this works. So here's just a diagram of a spike protein. This is for MHV strain JHM. I'm going to talk about initially. All coronavirus spike proteins have two subunits, S1 and S2. S1 contains the receptor binding domain, and S2 contains all the machinery for fusion and viral entry. So there have to be two cleavage events of spike before it can mediate fusion. One at this S1, S2 border, and one at S2 prime, which reveals a basic fusion peptide that's necessary for the actual fusion event. So some coronavirus spike proteins have a so-called furin site at the S1, S2 boundary, shown here. This JHM has a very basic, very good, very efficient furin cleavage site, shown here. And this comes out of the cell completely cleaved. So when entering a new cell, it enters by the direct plasma membrane route almost entirely. A closely related strain, MHV2, has a non-functional furin site. It has uncleaved spike, and it enters by the endosomal route because it needs to get both cleavages done here. Apparently, TMPRSS will do the second cleavage, but cathepsin is probably able to do both cleavages. And then we have A59, another related strain that has a less basic cleavage site. It's partially cleaved, and it actually enters by both routes. And the interesting thing to me is that all of these viruses are perfectly pathogenic. So whether it's cleaved by furin during production or not really has not a huge effect on the ability of the virus to cause a disease. So why do we care about this? Because it tells us something about antivirals. So an antiviral like chloroquine would only be expected to prevent entry by this route. Same for cathepsin inhibitor, whereas a protease inhibitor, for example, Camostat, would only inhibit a protease by this route. And so if the virus uses both routes of infection, we really have to think about what inhibitors are going to be worth using. And this is going to change really from cell to cell. While we know TMPRSS and ACE2 are the important molecules, or we think they are, for alveolar type 2 cells, we don't really know for all other types of cells possible. Okay, so I'm going to talk about transcription now. So here's the coronavirus genome, the 5' cap, 3' polyadenylated end. These yellow strips are transcriptional regulatory sequences, and they denote the beginning of each open reading frame and each messenger RNA. So the first thing that happens, as I said before, is a RDRP or RNA polymerase is synthesized or translated from the genome. It then transcribes from the 3' end a negative strand, a full-length antigenome, which then is transcribed or replicated back into full-length more genome RNA. At the same time, the polymerase transcribes subgenomic negative strand RNAs, and this is done by a really interesting, complicated process where the RNA is transcribed until here, and then the polymerase jumps with the nascent strand to here and makes this very small RNA. At some frequency, it may translocate from this TRS or this TRS, making all of these mRNAs. Then the subgenomic RNAs are transcribed back into full-length, into subgenomic messenger RNAs shown here in the blue. Okay, so what does this tell us? It tells us something about testing, for one thing, because here's a gel of messenger RNAs from, this is mouse hepatitis virus from 1983. You can see that there's that most abundant message is the smallest one, and they're sort of less and less abundant. And here's the genome. So a lot of the tests for, the diagnostic tests for SARS or for COVID amplify a piece of RNA within the replicase gene shown here. So this kind of amplification will detect genome only, whereas some tests also might, or actually do amplify in the nucleocapsid gene, and this would detect all of the mRNAs. So if the sample is containing only genome RNA, you'd expect really the same amount of amplification of both genes. However, if there's actively replicating virus, you would expect a lot more amplification of the N gene than the replicase. And this could actually be used, I think, to tell whether virus is replicating or not by amplifying both of these genes. Okay, so translation. So I showed these are all the mRNAs, and each of these is translated from its 5' end, usually into one protein, but sometimes occasionally two from overlapping reading frames shown here. But the really interesting translation occurs at this end. So this replicase gene that is translated into two proteins, PP1A and PP1A1B, which are then processed into 16 replicase proteins by viral encoded proteases, which I'll show in a minute. So there really needs to be, and how does this happen? There's actually a frameshift sequence in the RNA, in the genome RNA about here. So when the ribosome gets to the end of PP1A, it sort of stalls because of this structure, the pseudoknot structure, and moves back one frame, and then at about 20% frequency translates through to the end of ORF1B, making this PP1A1B. Okay, so let's look at these proteins a little bit more. Encoded in NSP3 is a papain-like protease that processes at these two sites. The then 3C-like protease processes the rest of these sites. This is the main protease. And then the RNA-dependent RNA polymerase is encoded in NSP12, and that replicates and transcribes RNAs. But there are really, and we've all heard about remdesivir, which is directed against the RDRP of probably any coronavirus. And then these protease inhibitors have been used against the 3C protease. I don't think they've really been that successful. This is a chymotrypsin-like protease, and these were HIV drugs. So in summary, there are proteases to process the proteins, and there are an RDRP to make the RNA. But there are really a whole lot of other conserved coronavirus proteins, and they have really interesting activities. These, the activator, the primase, the helicase, are all kind of auxiliary to the RDRP. They're part of the replication complex. There are 3 proteins involved in capping, this NSP16, NSP14 and NSP13 all contribute to capping the 5' end to protect the RNA from host cell responses. There's an endo-U, which I'll talk about a little later, that reduces double-stranded RNA accumulation, again, to prevent host responses to infection. And then as well, this XON is a proofreading enzyme that keeps the virus from becoming too attenuated by having too many mutations. And then the NSP1 and NSP3 also encode other multiple host antagonist activities. So, in summary, the enzymes promote synthesis and stability of RNAs by capping the 5' ends and protecting from host cell sensors and interferon response, which I'll talk about a little bit more later. So the coronavirus genome codes many how uses a large part of its coding capacity for host antagonist activities. And here's an example of how a conserved coronavirus domain can serve as a potential drug target, so that a drug that would be directed against one of them would probably work on all coronaviruses. So here's just an example. This is a model of the SARS coronavirus, the original SARS, NSP13, and predicted models for MERS and MHV, NSP13. And this molecule here fits into the binding pocket for all three of these enzymes. And you can see below, it also inhibits replication. We've plotted replication of virus against increasing amounts of compound. And you can see that each one of these viruses is inhibited by this with kind of similar kinetics. So in summary, many conserved features of coronaviruses, the subgenomic mRNAs, the non-continuous RNA synthesis, the ORF1A1B frame shifting, the proteases, the RDRP and RNA modifying enzymes, and further host antagonist activities. So from here, I'm going to kind of switch into the other part of the talk and talk about coronavirus antagonism of double-stranded RNA-induced antiviral pathways. So coronaviruses are really good at shutting down host innate immune responses. And here's an example for MERS. If we look at interferon lambda and interferon beta, this is in A549 respiratory-derived cells. You can see that there's barely any interferon induced, 10-fold at the most latent infection for lambda, and 5-fold to interferon beta. And just in contrast, this is for Sendai and Sinbis, two unrelated RNA viruses, and they induce, this is the log scale now, so they induce like 1,000-fold and 10,000 to 100,000-fold, and really high interferon stimulated genes shown here for OAS2. So we want to understand how this happens. So these are the double-strand activated pathways that we're concerned with. So viral double-stranded RNA is recognized by several host receptors, one of them being MDA5. Coronaviruses are recognized by MDA5 and not RIG-I, which signals through MAVs to produce interferon, type 1 and type 3, which signals using STAT transcription factors to induce interferon-stimulated genes shown here, which then reduce virus replication and restrict spread. But in addition to that pathway, we have this pathway that my lab has been very interested in working on this pathway for many years. Oligoadenylate synthetases perceive double-stranded RNA. They produce 2,5A, a small 2', 5' linked oligoadenylate that causes dimerization and activation of ribonuclease L, which leads to degradation of single-stranded RNA, which restricts virus replication, but also reduces protein synthesis and induces apoptosis and inflammasome activation. And the third pathway is PKR pathway. PKR also senses double-stranded RNA to autophosphorylate and then phosphorylate PEIF2 alpha, which is a protein synthesis initiation factor, which leads to reduction of protein synthesis. And this pathway, these two pathways are often thought of as sort of downstream pathways of interferon signaling because interferon upregulates OAS and PKR, further inducing these pathways. But actually, these pathways can be induced really independently of interferons. And that's probably why some viruses have specific inhibitors of this pathway. So the first thing I'm going to talk about, oh, and I should mention here that OAS RNA cell pathway is very similar in bats. We were interested in that because all these viruses have their origin in bats, and there's a really interesting question as to whether bat innate immunity contributes to their not getting sick from these viruses. So the first part, I'm going to talk about the coronavirus enzymes that specifically shut down this pathway. These are phosphodiesterases. They specifically cleave 2', 5' oligoadenylates. They're members of the 2H phosphoesterase family, which these two catalytic motifs, HXSTX, and they're found in the beta coronavirus lineage A, MHV and OC43, and in the MERS coronaviruses, and the toroviruses, which are related nidoviruses. They're all found in different proteins with each of these genomes, and they're not found in the SARS-like viruses. This is probably only structural protein among the different lineages that actually have some homology, the only non-structural protein. And in here, this case, it's expressed as part of the replicase, which is kind of curious. So how do these proteins work? So here's the NS2 from MHV. We're going to talk about mouse hepatitis virus, where we've studied this the most. And so we have two catalytic histidines. We got, this is a small protein. It's not essential for replication in tissue culture, nor in the central nervous system, which is one of the targets of mouse hepatitis virus. So we started with two viruses, a wild type virus and a mutant virus, in which this histidine is mutated to arginine, and this inactivates the enzymatic activity. These came originally from Stuart Siddell in Britain. So we looked at replication in bone marrow derived macrophages. So the really nice thing about this system is we can study a mouse virus in its natural host. It's a model, but at least it's in its natural host. And you can see that MHV replicates really well. This is a log scale, 10 to the 6 platforming units, whereas the mutant virus is really completely dead for replication. However, in RNA cell knockout cells, the virus replicates, recovers replication, the mutant as well as the wild type. So we wanted to understand why this happens, and so we looked at a couple stages of this pathway, and we quantified 2,5A using a FRET assay. So this is an indirect assay for 2,5A level, and we can see that while wild type virus generates not so much 2,5A, the mutant generates a lot more 2,5A, and as a consequence, it activates RNA cell, and we look at this by an RNA integrity assay, a so-called CHIP assay on a bioanalyzer, which generates a kind of a gel, a pseudogel, and you can see that 28NS ribosomal RNA are intact in the wild-type infected cells, but quite degraded in the mutant infected cells, suggesting or indicating that RNA cell has been activated. We looked at a similar kind of question in vivo in mice. So in these infections, we infect mice with about 5,000 platforming units. The virus replicates, peaking at day 5, and then is cleared by the T cell, primarily by the T cell system. So we looked here at amount of virus in the liver at day 5, which is the main target of the virus by this route of infection. We saw, again, very high titers of virus in the liver at 10 to the 7 platforming units, and really no mutant replication at all. Whereas in the knockout mice, mutant recovers. So this suggesting that this NS2 inhibitor is really potent. And then the same thing was true when we looked at liver sections stained for viral antigen from wild type virus-infected wild-type mice or RNA cell knockout mice. We see quite a lot of antigen, whereas for the mutant virus expresses antigen only in the knockout cells, nothing detectable in the wild-type. And the same when we looked at by H&E staining for pathology, there's really no destruction at all of the mutant virus in wild-type liver, but in the wild-type virus, irrespective of genotype, and in the mutant virus, in the mutant liver, we can see all this inflammatory activity here. And also destruction of the liver. So, okay, so NS2 has a major effect on this pathway, but another enzyme or another viral protein, the endo-U or the NSP15, also antagonizes RNA cells. So this is one of the replicase proteins that really has a somewhat similar activity, but different. So just to look at this a little bit, this is endo-U. We had a mutant with this mutation that inactivates its enzymatic activity. And this was obtained from Volker-Thiel, and we can see that virus replicates, wild type again replicates well in bone marrow derived macrophages, and mutant not very well. This is pretty similar to what we saw with the NS2 mutant, but there's a difference. One of the mechanism here, so Volker-Thiel and Susan Baker had shown that this endo-U mutant generates more double-stranded RNA than the wild type, implying that the wild type was preventing accumulation of double-stranded RNA. So in this experiment, we stained bone marrow macrophages from infected wild type and mutant infected MHV for both single-stranded RNA and double-stranded RNA. And we do see more double-stranded RNA. It's not quantified here, but there is more double-stranded RNA by other assays in the mutant-infected cells, but there's also a lot more of single-stranded RNA expressed, as you can see here. And in a paper that's just being revised for resubmission, we showed that actually the endo-U cleaves the genome RNA at its 3' end, so it sacrifices its own level of RNA to prevent the production of more double-stranded RNA. I think that's a pretty interesting mechanism. And again, here's the endo-U mutant. It activates RNA cell by this CHIP assay. Here's wild type and mutant. But it fails to recover replication in RNA cell knockout macrophages because it also activates, the double-stranded RNA also activates PKR, and we needed to use double PKR RNA cell double knockout infected macrophages to show that the the mutant virus would recover replication. So just to summarize this, so both the PDE and endo-U are required to antagonize activation of RNA cell. Double-strand RNA is perceived by OAS, but it's also limited in production by endo-U. The OAS synthesizes 2,5-A, which is knocked down by the 2'5' phosphodiesterases of MHV NS2. We also were able to substitute NS2 within the MHV virus backbone with the MERS protein or the rotavirus protein or a cellular phosphodiesterase, and all of them function to prevent this activity. RNA cell fails to dimerize and activate, and virus replication and pathogenesis can proceed. So I'm going to go on and talk a little bit about the MERS, two accessory proteins of MERS, NS4A and NS4B. NS4A is a double-stranded RNA binding protein, so as such would be expected to prevent activation of all three of these pathways, whereas NS4B is a 2H phosphoesterase with 2'5' phosphodiesterase like NS2, but it has a nuclear localization signal on it and also has the ability to prevent nuclear translocation of NF-kappa B. So we did show using an MHV kind of assay that this virus, for NS4B can serve as a RNA cell antagonist and can rescue replication of an NS2 MHV mutant. So it's fully active as an RNA cell antagonist. So with Ralph Barrick, we made mutants in NS4A and NS4B within the MERS genome. And we did a CHIP assay to look at activation of RNA cell. And we saw that when we mutate the enzymatic domain of the MERS NS4B, we get a small activation of RNA cell. There's no effect of mutating the NLS, which would be expected. We also made mutants of NS4A and NS4A4B, and we can see that the NS4AB mutant also activates RNA cells similarly to the catalytic mutant, but not much from the NS4A alone. And perhaps consistent with this, when we looked at PKR activation in this Western blot, here's PKR and PKR using Synbis virus as a control, we can see that Synbis virus really well phosphorylates PKR and PEIF2 alpha, whereas MERS coronavirus does neither of these, and the 4A mutant does autophosphorylate PKR, but really no detectable PEIF2 alpha. So again, mutating 4A or 4B causes a kind of weak activation of pathways. So we suspect that there's something else going on. The 4A, 4B mutant also allows the transcription of interferon lambda, beta, and interferon stimulated genes. You can see here, wild type really induces almost nothing, which I showed earlier, but the mutants induce somewhat more interferons and interferon stimulated genes, but still really dwarfed by something like Synbis virus, which is inducing 100,000-fold interferon lambda as opposed to 50-fold. So we believe that probably the endo-U, the NSP15 of MERS is also contributing to shutting down these pathways, and we're actually looking at that right now. So just in summary, the viral double-stranded RNA is perceived by OAS. We've shown that NS2 acts at this site here, and NS4B as well. And we showed that NS4A acts to prevent the activation of all of these pathways, and endo-U serves to downregulate all double-stranded RNA. Okay, so now I just want, the last few slides, I want to talk about our recent data on SARS. So here again are the 3' ends, the accessory proteins of lineage A and lineage C coronaviruses, and here's the lineage B. There's not, we don't see either a phosphodiesterase or a double-stranded RNA binding protein. The proteins that have been talked about in SARS-2 about possibly being interferon antagonists were ORF3b, which is truncated in SARS-2, and the ORF6 protein, which in SARS-1 interferes with nuclear translocation of STAT1. So we're really interested in these proteins now. So to study this, we first constructed A549 ACE2 expressing cells expressing the receptor, and you can see here the virus replicates in them really well. The green is the stain for nucleocapsid protein and this massive syncytia. This is all from spike on the surface of cells causing these very large multinucleated cells. And the virus replicates quite well. This is just 2 different clones, up to more than 6, almost 7 logs of virus, almost as well as you'd see in a viro cell. So we looked at induction of interferons and ISGs, and again, they were very poor. Here's Synbus virus inducing 2,000-fold interferon lambda, whereas SARS-CoV-2 much less, the same for interferon beta, and the same for OAS2. But surprisingly, SARS-2 actually does activate RNA cell, different from MERS. And here we have Synbus virus, which activates really well. And SARS-2, we can see some weaker activation. So we wanted to compare it to MERS. So we infected, these are CalU3 cells, another type of cell that both viruses can infect. And we saw here that there's, again, very little induction of interferons. But SARS does seem to induce ISGs better than MERS, shown here, and this is the MERS mutant. So both SARS and MERS are dwarfed by the amount of what the mutant can express, but it does look here that SARS-2 induces more interferons and certainly more ISGs than MERS does, although it doesn't replicate very well, shown here. And again, here we can compare the 2 viruses for activation of RNA cell. We can see that SARS-2 activates RNA cell weakly, MERS not at all, and it's MERS 4A, 4B mutant is very similar to SARS-2 shown here. And the same holds true for activation of PKR. Here's SARS-CoV-2 does phosphorylate PKR as MERS mutant, what MERS wild type does not. And the same thing, we also found that SARS-CoV-2 phosphorylates pSTAT, whereas MERS does not. So in summary, it does seem like SARS-CoV-2 may be not quite as adept as MERS at shutting down the host responses. So in summary, coronaviruses encode multiple interferon antagonists in conserved non-structural proteins and in lineage-specific accessory proteins. In the murine coronavirus model, both NS2 and ENDO-U are required to prevent activation of RNA cell. Expression of both proteins is required for replication and pathogenesis in the liver, but not in the brain. And I didn't really talk about that, but this is a organ-specific antagonist. And MERS-CoV-2 expresses 2 accessory proteins that, when deleted, results in weak activation of all 3 pathways. Similarly, inactivation of ENDO-U, I didn't show this, but inactivation of ENDO-U had a small effect on these pathways, and we're now looking at a double mutant of ENDO-U and NS4A, which we think will result in activation. SARS-CoV-2, like MERS, weakly induces interferon. However, SARS activates RNA cell and induces phosphorylation of PKR and STAT, suggesting it's not as adept as MERS in shutting down these pathways. And what we're doing now is we've made we're constructing mutants in some of the putative or predicted interferon antagonists, and we've also constructed A549 cells in which we have knocked out some of the genes like PKR and RNA cell to see the effects of knocking out whether these pathways are really restricting replication of the virus or not. And this is, I want to acknowledge everybody in my lab. The people that are bolded here are actually working very hard in the BSL-3 on SARS-CoV-2. Everybody else is working hard supporting them. And these are past members in my lab who were really important for setting up a lot of this, a lot of the systems we work with. And these are our collaborators and our funding from NIH. and I will stop there for questions. Oops.
All right, that was great. Yeah, thanks so much. Maybe I'll just start with a few while we wait for questions to start coming in on the chat. So maybe following the order of your talk, at the beginning you have that very nice slide about the history of coronaviruses and the different eras. Yeah, I was wondering, what your thoughts on why this pathogenic coronavirus era begins around the year 2000, whether those other four more benign human coronaviruses might have started the same way as what we're seeing right now with SARS-2.
Well, I think they may have started the same way. I think that there are precursors to some of them in bats, but I think like for OC43 is believed to have evolved probably from bovine coronavirus. So it probably has its origins and may have its origins in rodents rather than bats, whereas some of the other ones do have probably have precursor or parent or ancestral viruses in bats as well. Why 2000? I think that my thought is that there probably were other spillovers as well, but maybe didn't cause epidemics. It's hard for me to believe that there are only ever 3 spillovers.
Yeah, it's interesting.
And actually, somebody was telling me that in that people, I think in Southeast Asia, they find in people that don't have SARS T cells with specificities against non-structural proteins, which are the conserved proteins. So there could have been stuff we don't know about.
Interesting. And you showed us elegantly how the virus dismantles some of the sensing pathway and interferon responses. So from a therapeutic standpoint, if you were to give interferon early in infection, do you think that would have a role in therapeutics?
Well, that's been actually, maybe bandied about a lot. And there's a really interesting paper from Stanley Perelman's lab where he did, it's an animal model for SARS where he showed that actually. He showed that if you give interferon early, you can wipe the virus out, whereas late it actually really does, it is pathogenic. So yeah, and I've heard talk about people, I think there might even be clinical trials using interferon.
Yeah, I know of one where it's part of a 3-drug regimen. There was a paper where they combine it with the HIV protease inhibitors and
yeah, I don't think the protease inhibitors.
I know.
Yeah.
It made me think that interferon really could be quite important because they claimed a very strong effect, and yet they were combining it with drugs that shouldn't be active. Could you comment a little bit about how MERS versus SARS-CoV-2, the differences that you've described regarding host defense might explain differences in how they spread and cause disease in humans?
We really don't know. This is very early data, and it's so micro. I think we need to know what goes on in the nose. It seems to be like probably SARS lingers longer in the nose, and that's why it's spreading pre-symptomatically or pre-really bad symptoms. I don't know. I mean, this early data would suggest to be more benign than MERS-CoV. But again, it's still not inducing very much interferon. If you look at a big scale, like I was showing, if you look at Sinbis, they're all producing nothing compared to that. The fact that MERS produces a little bit more, I'm not sure how significant that is.
Yeah, and just on the subject of MERS, I think I remember when you were giving the seminar before the whole, when we were just getting going, that there was some question at the time about the intermediate host, and for MERS, it's the camel. Do you have any thoughts currently on whether this SARS-CoV-2 jumped from bat to human, or whether?
I haven't heard nothing really new. The pangolin, I think, is kind of out of favor. It's certainly not been proved to be so nothing really matches. I heard a very interesting story that there's apparently an obscure paper that I haven't seen yet. Somebody in my lab was telling me about, which suggests that maybe this that a similar virus, not this one, had passage directly from bats to humans. So we don't really know. It could have gone direct. By precedent, we say no, but we don't really know.
And then there's a question I was also wondering about: how do the RNA sensing pathways vary with aging? Could they explain some of the differences we see medically and clinically in people of different age groups?
I don't know how the RNA sensing pathways change with aging, if they do. I'm trying to think. I mean, activating these pathways is bad in late disease. So I don't know that it's like, it would almost suggest you had more sensitive pathways in older people, but so I don't know.
Yeah, I was wondering a little bit while you were talking about cell-to-cell communication and whether any of the mediators or even antiviral proteins that the virus can spread through gap junctions from cell to cell to kind of hit a
Well,
neighborhood cell.
well, you know, the virus spreads really well cell to cell in tissue culture. You know, we've studied this a lot in MHV. You don't see so much cell to cell fusion in MHV, but it certainly spreads like you saw in that. I was amazed when I saw it, but it doesn't do that in vero cells. It's really interesting. I mean, it really depends.
You know, there's some evidence that CGAS products can spread from cell to cell through gap junctions.
Yeah.
Alert a neighboring cell. So I was sort of wondering, is there a corollary here where
2,5A
2,5A can spread, or even the coronavirus protein against it?
I really don't know. I don't know.
Sure. So we have a nice question about what you mentioned with work on comparing actual transcript of the virus to tell what cell is actually infected versus this phenomenon where you might pick up viral RNAs in PCR tests and that may not reflect active virus. And so it's a question asking whether you know of any proof of concept for coronavirus where this has been used medically, sort of get a sense of active coronavirus replication in a clinical test.
I mean, I do think that, you know, it's very complicated. First of all, like there's probably, we just measured this, there's something like 1,000 genomes per infectious unit. So a lot of so first of all, the test for genomes is way more sensitive than the test for virus. And we found that we can't, this may not be exactly what you're saying, but this is all relevant and tied together. But when you try to isolate virus from nasal nasal tests, we found that if you have less than 10 to the 5 genomes, you can't culture it out. They're pretty sensitive tests. So there's that. And then I don't really know when you're testing whether you're looking at genomes or replicating virus or both. It's really kind of confused in my mind, and nobody seems to really differentiate that much. But if you were to amplify both genome and message there's so much more message than genome that if you amplify N, you should get hundreds of times more if you're really having replicating virus. So that could be used as some kind of diagnostic. And then does it matter? Does it really, like if you find one plaque-forming unit versus it's about sensitivity. So I really don't know. When people say that someone's recovered and they still have a positive for RNA, I'm not really sure what that means. It's really confusing to me.
That's where that would be very usel. Or the test that you've described, where you could look at processed RNAs and things.
You could, but then maybe you have there's all kinds of possibilities. You could have remnants of message too. It's just hard to really know. I
see. Well, do you think that you could leverage some of this viral evasion that you've discovered in vaccine strategies to make some sort of an attenuated strain where this is disrupted, or do you think that recombination that happens so often in coronavirus would be too high of a risk?
I think there are a lot of ways to make attenuated viruses. People have talk about using, if you mutate NSP14, the proofreading enzyme, it really screws up a lot. I mean, it screws up recombination as well as polymerase jumping, and there's a really interesting paper on that. People have done things like in some of these viruses, you can get rid of the E protein, which sounds crazy. It's a structural protein. So you get a highly attenuated virus. Luis and Juanes has used that strategy. You can also rewire these things. So you move genes around and you attenuate. So there's lots of ways to attenuate, but there's always that worry of recombination. I like the idea of attenuated vaccine, but I think nobody else is using it because it's possibly dangerous.
I guess we'll have something this winter that we haven't really had before, but do you think there will be some recombination happening between this coronavirus, the SARS-2 virus, and the other 4 circulating?
Okay, I've thought about that a lot, and I've asked colleagues, and we all think no, but we don't really know. They're so different. I mean, they're really different. I don't know how much homology you need to recombine. But they're in such different lineages. Maybe SARS-1 and SARS-2 would probably recombine, but that's not a possibility.
So we have a question, just something that you mentioned, where there's been these cycles of intense interest in coronavirus research after SARS-1 and MERS, and then this drop off, and then we're having the interest again. So do you think that there's some way to improve our ability to stimulate research in areas before they become pandemic critical?
Well, I mean, it would have been nice. I think part of it, you know, if there'd been more funding for corona, I mean, people just could have yawned when they thought about coronaviruses. Before SARS, like people thought it was, one of the reasons I went to work on it was my insecurity and wanting to work on something that was not going to, after coming out of Bishop and Varmus, I didn't want to work in something super competitive. And I found this thing that looked wide open.
Yeah.
And then nobody really cared until SARS. And then there was money pumped in, and then there was money, not so much.
Yeah.
We'll see what happens now.
Yeah,
I don't know. You know, a lot of people rushed in now, and I don't think they'll rush out as fast as in the past, because obviously this is much bigger. But I'm sure that it's going to settle out. People that don't get funded are going to disperse.
Yeah, sure. And so we have a question about, you mentioned this nuclear localization signal and on one of the viral proteins, and can you comment a bit more on the function of?
Okay, so that's interesting. We're very interested in that. So NS4B, I mean, NS4B and also ORF6, well, NS4B goes into the nucleus, and it's believed that in addition to being a PDE, phosphodiesterase, it prevents some translocation of transcription factors, for example, NF-kappa B. And that's something, there's one paper on it, we're working on that as well. I think it's really interesting. And then also the ORF6, though, by a different mechanism, they both have to do with, what do you call it? The enzymes that, or the proteins that transfer into the nucleus. Importance, sorry, importance.
Carrier-afferents,
carrier-afferent, sorry. And ORF6 also prevents translocation of STAT1 in SARS-1, and we're very interested in seeing if that's the case with SARS-2, because the ORF6 are slightly different, and we do see phosphorylation of STAT. Whether it goes in the nucleus, I don't know yet. But so I think both of them, you know, mostly RNA viruses don't go into the nucleus at all, except for flu, that's infamous. But many viruses now we're finding do have some protein that goes into the nucleus and that interferes with things like splicing and other, you know, other cellular events. Yeah, viruses are really diabolical. They have so many, even these little, I mean, they're small viruses, even, you know, they only have 7 or 8 genes.
Yeah, no, we had the last talk in the series was from Craig Weiland on screen in viro cells for host factors that important
for SARS.
Yeah, SARS-2, looking at things that were protective or sensitizing towards the cytopathic effect. And a lot of nuclear proteins showed up in his hit list, but maybe they're driving the expression of something in the cytosol that was clear.
Yeah, and viro cells don't have this interferon response. You're kind of eliminating that whole. Yeah, I could see you could do that on purpose, maybe to look at other things other than interferon.
And so I was wondering also about, you mentioned in one of the earlier slides that MDA5 is sort of important for sensing these, but not
interesting, right?
But not Reg I. And I was wondering a little bit about. I know they have differences in the sizes of RNAs they sense. Is there other knowledge of why MDA5 and not Reg I?
We found that like really a long time ago with MHV. But that's true for other plus strand RNA viruses also, like picornaviruses. And so I don't know why, because you'd think they'd make every kind of RNA that you can imagine, because I mean, they have plus strand, they have minus strand, they have capped, they have uncapped, they have double strand. I don't know, it's really mysterious. So that's one thing we'd really like to know is why, you know, what about this, what actually is sensed by MDA5? Because on a completely other project, which I did talk about last time for ADAR knockout. So it turns out that endogenous double-stranded RNA is also probably sensed primarily by MDA5 and not Reg I, which is sort of
that is
whatever an ADAR is sensing double-stranded. Similar RNAs to ADAR and MDA5. So somebody should, I mean, in my dreams, just like look at, you know, all the sensors. I think people are doing that, but we really don't have a very good idea.
Yeah, that's interesting. And I mean, do you think that this ability of these coronaviruses to evade the interferon pathways has something to do with the lack of durability and the memory to them, such that we're still waiting to find out about SARS-2, but for the other ones, there tends to be a shorter-lived memory.
Ooh, I have no idea.
Is that tied to the interferon evasion somehow?
I don't think it's interferon. It's more probably B cells and T cells.
Yeah.
But I don't know. That's interesting. Yeah. I mean, all viruses have interferon antagonists, and then I always think too, like Synbis and Sendai that I showed, they replicate really well despite the fact that they activate all these pathways. So it's really, it's also very much cell context. Like RNA cell is not even activated in some cell types because OAS levels are too low. They have to be really high, like in myeloid cells, primarily endothelial cells.
Yeah.
So it's really cell type dependent. And so, like, as I said, if you did the same infection and looked in the brain, you would see absolutely no difference between that NS2 mutant and the wild type. It's only in an organ that has a good interferon response, a good level of interferon stimulated genes.
Mm-hmm, mm-hmm. Good. That's interesting. And then we have a question on, you mentioned 10 to the 5th genomes in certain cultured cells. Is there a sense of how many of these are intact versus and sort of non-mutated versus?
You mean how many are intact?
Yeah, yeah. I know there's the XOM, the proofreading activity that helps to.
Oh, okay, okay. So
Yeah.
I'm sure there's a lot of intact genome. It's making all these proteins and it makes, it's very productive infection. I mean, it's really a kind of curious thing that we think that's what's going on, that we know that. So we did this sequencing project. David Barton really did most of it. We were collaborators, and he showed that that the endo-U cleaves all along the genome, and it has a major slot near the 3' end. So we just, we think that we suggest that maybe it's just cleaving itself there to prevent making more double-stranded RNA. But I mean, the observation is there's less double and less single-stranded RNA in the wild type versus the endo-U mutants. So it's really, to me, it's really interesting. So all the viruses have endo-U, but then they need to have these other little, fine-tuning proteins to really shut down pathways. So they must really need to shut them down. It must have an important function, but I'm not really sure what it is.
That's interesting. And sort of the last question is, this seminar series that we have is also focused on the microbiome. And so you've had the ability to study MHV68 in the mouse. Have you ever come across microbiome?
All right, there's a problem here. It's not MHV68.
That's I got the wrong one.
MHV68 is a norovirus, I think, right?
Okay, got the wrong one. Yeah, but for murine hepatitis virus.
We have not done that, but I'm telling you that ever since this SARS, I have gotten requests from, I don't know, 50 people wanting MHV, and one person or one or two people said they were going to look at microbiomes. So we haven't done that, but you could do that.
Okay.
Yeah, you could do. And a couple of people from Harvard actually were interested in MHV.
yeah, it could be interesting, just given the different disease and different age groups and populations. Could be.
Yeah.
Good. Well, that's great. So thanks so much for.
Hey, you're welcome.
For taking
this. Is fun.
Today. Yeah, appreciate it.
A lot of questions. Okay.
Okay, great. And so everyone, just a reminder, we have one more of these next week with Pardeep Sabeti's group next week at 9 o'clock.